Alright, hey everybody, so sorry about the delay on this one, but here it is. This is going to be the chapter 11 review video, and this is going to be all about cell communication and signaling. This should be a little bit of a quicker video, but there is a couple very dense things. So cell communication, it's very broad, and that's why there's a lot of little things to cover in the chapter. But let's just get into it. We are going to have three large categories of communication between cells. We're going to have direct contact, which you can think of as your neighbor. You're going to have local signaling, which you can kind of think of as sort of your town or the city you live in. And we're going to have endocrine signaling, which is going to be long distance. So that's going to be, you're sending a letter to someone far away. And when we think about this in terms of cells, a neighboring cell is going to be sort of same cell type. Local signaling is sort of going to be within the same system. And then endocrine signaling is going to allow systems to communicate, such as the nervous system, eliciting the sending of epinephrine to the heart would be an example of that. Those are the three large categories. We're just going to go one by one. So first we have direct contact. This first one's going to be really, really easy. Direct contact is going to be facilitated by cell junctions. And remember, direct contact are those neighboring cells. So this is going to be one cell communicating something to the cell right next to it. And I have a little drawing here. So this is two cells. We'll call these epithelial cells, and they're attached to a basal lamina or a basement membrane. You don't need to know any of that though. But what we are looking at is these three sort of junctions right here. And these are the three types of junctions that for this class that we want you to know. There are a couple other ones that we won't go into. You'll get more into that into cellular biology classes. But at the top, we have these two proteins. We have one on each cell, so we have a protein here and a protein here. This protein belongs to this cell, this one to this cell. But on this top one, we have these two proteins, and when they come together, they sort of form like a channel. So you can imagine my two fingers are parts of the channel, and when they connect together, there's this space in between them. And that's sort of this space right through here. And you can see I drew these dots moving through because this structure is allowing molecules to go from one cell to the other cell. And this is going to be called a gap junction. And they are really good at transporting molecules. Or it could be ions too, So that is going to be a gap junction. You can remember a gap junction, it's literally creating this gap for things to move through. And it never has to exit the cell. You'll see that when this molecule goes from here to here, it never has to go into the extracellular fluid or the extracellular matrix. So that's gap junctions. And the next two I'm going to lump together because as far as this course is concerned, they do the same thing. It's tight junctions and desmosomes. And these are really good at just anchoring cells together so they can sort of mesh together and sort of hold them together like that. And that would be more like a desmosome. And then your tight junctions are going to really be good at stopping things from going through. You can imagine something wanting to go down into this sort of basal lamina area. That's really good for stopping that. So that's it for direct contact. Really simple. Next up, we get a little more complex with local signaling, which is going to be facilitated by paracrine signaling. Don't get paracrine mixed up with endocrine. And later, sometime down the way, we're going to add exocrine. So there's a lot of these CRINE words. But paracrine is going to be local signaling. So paracrine signaling is going to be cells releasing either growth factors, hormones, cytokines, or neurotransmitters to other nearby cells. So I have two examples of this. Here we have a cell that is chock full of these growth factors. And growth factors are really good at telling cells to grow, split, proliferate. And this cell is chock full of them, right? And you have all these other cells, I've already drawn them in, that need more growth factor, especially like you have this little guy down here who needs a lot of growth factors. So what we can do is we can package the growth factors from this cell up in secretory vesicles. Remember vesicles, we went over those when we talked about sort of the organelles. So we can package them in vesicles, and through that process of exocytosis, remember exocytosis, exiting the cell, we can send these vesicles containing growth factors out, and they can start being picked up by other cells. And when that happens, then those cells can finally sort of proliferate and do their thing. And that's going to be paracrine signaling. Sort of a subdivision of paracrine signaling over here is going to be synaptic transmission. If you've heard the word synapse refers to the space between the end of one of your neurons and another cell. And that other cell is usually either going to be a muscle cell or just another neuron. So here I'm using a neuron, which is this structure, and then a generic muscle structure. And so this synapse is going to also be referred to as the neuromuscular junction. So synaptic transmission is just paracrine signaling using neurotransmitters. So neurotransmitters are just these molecules. A lot of times they are similar to hormones, which we'll get into, and they can be a lot of things. You know, they could be ions, they could be, acetylcholine. A lot of medicine kind of relates to this stuff. But anyway, we are going to have an electrical signal, because it's a neuron, and remember the nervous system runs on electrical signals. An electrical signal is going to elicit the release of neurotransmitters from this neuron, from the end of this neuron, into this synapse. And you can see there's not much space here between the neuron and the muscle, and that's very intentional. It's because then when we release these neurotransmitters, they can very quickly and very easily affect this cell by binding on receptors, or maybe they'll actually go through. But anyway, that's synaptic transmission. And that's going to be it for local signaling. So now, before we move on to endocrine signaling, I just want to briefly talk about hormones, which is a word that you have probably heard before. So hormone, by definition, is a signaling ligand that binds specific receptors on target cells, causing conformational changes to start a signal. Let's break that definition down just a little bit. So ligands, I sort of repeat a definition here because ligands are just signaling molecules. So anytime you see the word ligand, think signal. It's going to bind to a receptor and it's going to start some kind of signal or stop a signal. But anyway, so hormones have two different types. We have lipid soluble and lipid insoluble. And so if we think about what we know about the cell membrane, it's that phospholipid bilayer. And remember, we have these lipid tails on the inside and phosphate heads on the extracellular face and the intracellular face. So if I have a lipid soluble hormone, if it's able to dissolve through the lipid portion, that means it could go in the cell. It can go right through the cell membrane and bind to an intracellular receptor. And remember, lipid soluble if it can be dissolved in the lipids, that means it can go through. So lipid soluble can bind intracellular receptors, but could a lipid insoluble hormone get through the bilayer? And the answer is no. It can get here, and then it can't get through. It cannot dissolve through the lipids. So lipid insoluble hormones have to bind to extracellular receptors that oftentimes are going to be transmembrane receptors, which means they span the entire membrane. So you can see this receptor here, this will bind here, but this goes into the cell, so we can still get that intracellular response. So those are hormones. That's a super important word to know. They're going to be really important, and we'll go over a couple specific hormones. You don't need to know many, though. So I was going to try to draw out the G protein coupled receptor, but I think this is more of a whiteboard situation. So let me switch out what we're doing here. Sorry for the pause, but I gotta switch the setup around a little bit. Okay, so is this gonna work? All right, so now we are going to be talking about a specific receptor pathway, and we're going to be talking about the epinephrine pathway. This is really a very good sort of hallmark example of G protein coupled receptors. So the epinephrine pathway is going to utilize the GPCR, which again is the G protein coupled receptor. So if I have a G protein coupled receptor, it makes sense that I also will have a G protein intracellularly. So here we are going to have epinephrine. Now, epinephrine is a lipid-insoluble hormone, which means it cannot pass through this cell membrane, but what it can do is specifically bind to epinephrine receptors, which are GPCRs. I will say G protein coupled receptors, there are a ton of different ones. I just wrote it generally, you don't need to know, but know that the epinephrine will specifically bind to the epinephrine G protein coupled receptor. So when this binds here to this little binding site, it's going to activate this G protein coupled receptor. So it's going to go from inactive to active. That didn't write super well, but now it's active because we have binded this. And the reason it becomes active is because of a conformational change. And all that means is when it binds this ligand, which is epinephrine, it's going to slightly change the structure of the actual receptor, and that slight change in structure is what's going to make a response happen. So in the case of GPCRs, when they become active, they are going to bind to their respective G protein. So G proteins are a type of GTPase, which we've talked a lot about ATP. So ATPases, remember, like to cleave ATP and make ADP. G proteins do the same thing, but just with GDP and GTP, which is guanine, which is just a different nucleic acid. So you can see we have this inactive G protein, which is binded to GDP. So when this epinephrine binds and causes a conformational change in this receptor, it's going to cause this G protein to sort of go towards the GPCR, and it is going to exchange its GDP for GTP. And when it changes to GTP, that activates the G protein. So the G protein is going to sort of go over here, it's going to interact with the GPCR, and it's going to drop its GDP, right? So it's dropping that GDP, guanine diphosphate, and it's going to pick up a GTP, which is going to activate it. So this G protein is now active. When this G protein becomes active with its GTP, it is going to move downstream, and downstream just means we're moving towards the next step. We're moving towards the end of the pathway. If I were talking upstream, that would be referring to an earlier step. So we could say that epinephrine binding to the GPCR would be upstream of the G protein activation. And then, like we're saying now, the G protein is going to move downstream, so closer to our endpoint, and it is going to move to, hold on, I've got to scooch you again. It is going to move towards this adenylyl cyclase. So adenylyl cyclase is an enzyme that, when this G protein settles in here, so we have our G protein with our GTP. When this happens, it's going to activate the adenylyl cyclase. So it was inactive, but now it's active. And a couple things are going to happen now. The first thing is this G protein is going to cleave a phosphate off of its GTP. And when we cleave a phosphate off of GTP, we go back to GDP. And it's going to use that phosphate to sort of activate this adenylyl cyclase. So now that this G protein has been inactivated because it's back to GDP, which is its inactive form, this G protein can just go back and start the pathway again if it needs to. It can go back to its resting state that we had at the beginning. But now we have an active adenylyl cyclase. And what adenylyl cyclase does is it takes ATP and it converts it to a molecule called cyclic AMP. So we've talked about ATP and we've talked about ADP. AMP is just going to be adenosine monophosphate. And C is cyclic. So this is called cyclic AMP. You can call it CAMP if you want. So this is going to end the first stage of this signal being received, and this was signal reception. So right here, all of this, everything that we've done up to this point has been, can you see it up there? I cannot see it. Has been reception. So we're going to move into the second portion, which is going to be, I'll write it down here, transduction. Okay, so transduction is going to start with ATP going to cyclic AMP. And this is going to start what is called a signal transduction cascade. And it's called a cascade because it's just going to keep cascading, as you can kind of see. And there are more steps than I've written out here. But basically, the concept of signal transduction is that we are amplifying the signal. So as we move down this, we are going to amplify the signal. We're going to create more and more messengers. Scooch a little bit. So we start with one molecule of epinephrine, but as we go through this process, we start at each stage getting more and more signaling molecules. And that's because we have this cascade effect. So let's get into it. So we have this, kind of smudged it, didn't I? We have this inactive protein kinase A, or PKA. And what cyclic AMP is going to do is it's going to donate a phosphate, it's going to phosphorylate the kinase to activate the kinase. And real quick, a couple definitions that are good to know. Kinase, anytime you see the word kinase, that means that it is going to transfer phosphates. So it is going to do the phosphorylating. It is going to, kinases phosphorylate something. And then the other one would be phosphatase. So phosphatases, anytime you see like protein phosphatase, that means that they are going to take take phosphates away. So a kinase would donate a phosphate, usually by getting it from ATP, and then phosphatases take a phosphate, dephosphorylate. So that's just a couple quick definitions as we go through this. Okay, sorry, I'm gonna just one last time. There we go. All right. So we have ATP that has been converted to cyclic AMP, and that cyclic AMP is activating protein kinase A. So this PKA is now activated. So that activated protein kinase A is then going to be able to phosphorylate another couple of these molecules, which are phosphorylase kinases. So this is an inactive version, and remember, kinase is going to phosphorylate it into an active PKB, we'll call it, protein kinase B, or phosphorylate kinase B. But it's important to understand that it could do this to multiple of these. You could have a whole bunch of things, and this activated kinase can phosphorylate multiple different things. And that's why we get signal amplification, because this one can, let's say, phosphorylate 3, and then each one of these 3 could do that to another 3. And then it just goes on, and you can see how that signal could really get amplified by that. So then once we have this active PKB, it is going to activate by phosphorylating an inactivated glycogen, I believe it's phosphatase, into an active glycogen phosphatase. And when that becomes active, this is finally going to be our cell response. This active glycogen phosphatase is going to convert glycogen to glucose 1-phosphate, which can then convert into glucose. So that is sort of the whole pathway. And if you're a little confused by that whole pathway, dropped you, so sorry. That's okay. Look it over a few times. It's a difficult pathway. I have a little note sheet that sort of generalizes, and this is from a year ago, so this is old stuff, that sort of generalizes the process of transduction specifically. So ignore everything up here, but basically we have this inactive PK1 that goes to the active PK1, which activates the next one, which when that becomes active can activate the next one, and it just goes on and on like that. And as we do that, we get amplification through this phosphorylation cascade. So that was the epinephrine pathway. And so I'm kind of just going to write down a couple notes about that. Oh, let me grab the pen. So we're going to ignore all that. So in that epinephrine pathway, though, we had a couple molecules to define. So our primary messenger, messenger? I might have spelled that. Uh-oh. Let me grab a new pen. Sorry about that. My pen died on me. So we had our primary messenger, which was epinephrine. And then we also had a really important secondary messenger. And that secondary messenger was cyclic AMP. So secondary messengers in these GPCR pathways are usually going to be sort of the product of a membrane enzyme, if that makes sense. So sometimes it's calcium ions. Cyclic AMP is the one that we really want you to know, though, in this epinephrine pathway. So we had our primary messenger of epinephrine, our secondary messenger of cyclic AMP. And these words are very important to know, primary and secondary. You might be asked to identify what's a primary and what's a secondary messenger. Just know primary messenger is going to be the initial binding. That's usually going to be what the primary messenger is. So that's going to be a hormone, maybe it's a ligand, it could be a cytokine and paracrine signaling, but primary and secondary messengers, very important. Okay, so a couple general notes, well, one sort of general note here. Let me... Sorry, it's sort of cutting off a little bit there. There we go. So signals can elicit different responses depending on the cell type or receptor type. So what I mean by that is if I send epinephrine to a muscle cell and I send epinephrine to the heart, that's going to do two different things. Once epinephrine gets to the heart, it might end up increasing our heart rate, right? But when I send that same stuff to the muscles, it might make them more excitatory. So it might make them a little more on edge, so to speak. And that's just because those are different cell types, or maybe they're different receptors. So that's super important to know, is that one signal molecule, like epinephrine, can affect different cells in different ways. Most all responses that you get in any signal are either going to be a change in gene expression or a protein modification. All right, so that's just sort of a side note. Next, we're going to get into receptor tyrosine kinases, or RTKs. These ones are much easier, hopefully. So a RTK consists of two subunits here. And these are going to be transmembrane proteins, and I guess I should label this as intra and extracellular. So you'll see they have two binding sites, though, because RTKs can't just work as one subunit. They need each other. So they both need to bind a ligand. So once both of these subunits of this RTK have bound to its ligand, they are going to do what is called dimerization. And you may have heard the word monomer. So a monomer is one, a dimer just means two. So dimerization means these two subunits are going to come together with their bound ligands into one subunit. So now we have one unit when we had two before, and that's dimerization. So the next thing that they're going to do is autophosphorylation. And that means that these tyrosines, there are 3 tyrosines on each of these, they are actually it's going to be cross-phosphorylating. These two subunits that have dimerized are going to phosphorylate each other by taking 6 ATP and taking away some phosphates to 6 ADP. So now we have phosphates on the ends of all these tyrosines, and there is another GTPase called RAS that very, very commonly can come. And remember, RAS and any G protein or GTPase, when it has GDP, is inactive. And then up here, when it gets that GTP, it's going to be active. So how's it going to go from GDP to GTP? It's going to pick up these phosphates. And you can see again how this is going to be able to amplify a signal, because we started with these 2 ligands, but now we are able to get a total of 6 responses from these GTPases. I mean, there are other things that could probably interact with these phosphates as well, but RAS is a really good example. And then once that GTPase RAS is active, it can go down here and it can use a phosphate from this GTP to start phosphorylating inactive kinases, which can activate them, and then they can phosphorylate another inactive kinases, and it just keeps going through that signal transduction. Okay, and sort of the last receptor example is going to be ligand-gated ion channels. These ones are the easiest. So here we have intracellular, extracellular, and this is a ligand-gated channel. We'll call it a ligand-gated sodium channel because I drew sodium. So when this ligand binds to this receptor site, you can see there's almost this closed gate, or the closed conformation of the protein, stopping these sodiums from getting through. They can't get through. But when this ligand binds, it's going to cause a conformational change and open the gates and just let sodium through. So that's ligand-gated ion channels. I will show this again. This is my note sheet from a year ago, and it is relatively confusing, but if it helps anyone, it helps anyone. It's short. It sort of summarizes GPCRs, RTKs, and ligand-gated channels, and then how transduction works. Again, it's very jumbled. It's a year old, as you can tell from all the coffee stains and stuff. But if it helps, it helps. So there's that. Moving on, and sort of the last thing I want to talk about, we already talked about the epinephrine pathway. is going to be glucose homeostasis. So glucose homeostasis, I'm not going to go through the actual like cellular pathways. I just want you to know how it works. So what I mean when I say glucose homeostasis is regulating blood sugar. That's what that is. And this is going to be controlled by two hormones. And you've probably definitely heard of insulin, and maybe you've heard of glucagon. So what insulin is going to do, insulin is going to be released. So we are going to send insulin out into the bloodstream when our blood sugar levels get too high. And what that's going to do, remember, blood sugar means sugar in the blood, not in the cells. So insulin is going to bind to cells, and it's going to make those cells start taking glucose out of the blood, which is going to lower your blood sugar. So insulin is for when blood glucose is high, and it's going to cause glucose uptake by the cells. And when I say the cells, I mean like your muscle cells maybe will start taking up glucose. It could be any cells that insulin binds to. So insulin binds to a cell, and that cell then will take up glucose. However, that's when glucose is too high. What about when it's too low? So let's say our blood sugar drops really, really low. We were really, really physically active. We used a lot of energy. Then glucagon is going to be released. So glucagon is released when blood glucose is too low. So we now need more we now need more sugar in our blood. How are we going to get that? It's going to release glycogen, not glucose technically, I think, right? It's going to release glycogen from the liver, which glycogen breaks down into glucose. So this is also going to sort of bring up the point of signal termination is very important. You can see how if insulin stays active for too long, it could be a problem, or if glucagon stays active for too long, it could be a problem. So it's very important to be able to regulate when these signals stop too. Because let's say that I have really high blood sugar and then I immediately burn a lot of energy by sprinting. I don't want insulin to keep doing its thing endlessly, because then my blood sugar is going to be too low and other cells won't be able to get that energy. So I need to stop insulin and start glucagon at that point. So there's sort of this cycle where we upregulate and then we downregulate, and it kind of almost looks sinusoidal, or like, you might remember this from math class, but blood sugar goes high and then it'll go back down, maybe it'll go too low, and then glucagon will bring it back up, and then insulin will bring it back down. So these would be controlled by insulin. These sort of anything where it's getting high is going to be insulin is going to come in and lower it. And then when it gets too low, then glucagon is going to work here to bring it back up. I hope that makes sense. And then one super quick thing to mention is apoptosis. Apoptosis is programmed cell death. This is totally just this kind of a random little thing, but it's in the chapter. We'll go way more into apoptosis in another chapter. Just know that apoptosis is programmed cell death when the cell senses damage or an external death signal. This is very common with cancerous cells, and this is one of the big checkpoints we have to stop cancer in our bodies. And what apoptosis is going to do is it's going to receive either an internal or external signal, and then it's going to activate caspases and nucleases, which are just essentially enzymes that are going to start eating stuff. They're going to start eating stuff. There's going to be a whole process, but we'll go more into that in another chapter. So that is it for chapter 11, and that's it for the exam 2 materials. I'm releasing this on Monday, so a day before the test. Again, I apologize. This last video, I didn't have much time to get in. I was away for the weekend. But please remember that I will be in the lecture hall at 8 a.m. tomorrow, so an hour before your individual exam. please stop by, ask me any questions. We can go over pathways, we can go over exam questions from the collaborative, anything you need. So anyway, have a good night, good luck studying, and I will see you tomorrow.